Hi, my name is Dr Alikhani. I am Dean and Professor at CTOR Academy, and I'd like to welcome you to another session of CTOR channel. Today, as part of the biology of orthodontics, we are going to follow the discussion on the saturation point. If you remember from the previous discussion, we said in animal studies demonstrated that the rate of tooth movement in animals cannot increase by increasing the magnitude of the force, and that was due to a saturation of biological response, means that after that the biological response cannot be increased. That was related to maximizing the amount of the cell-free zone inside the PDL. However, does saturation point exist in human? And if it does, is it different between different humans? To explore this concept, we need to do the pilot study in human. What variable we use to differentiate between the humans? We can use ethnicity, we can use gender. We decided to go with the age because our previous study demonstrated that the age difference can demonstrate difference in biological activities. So let's do a pilot study where we get 16 adult and 16 adolescents, and we expose them to different magnitude of the force. So in each one of the group, we divide equally and randomly the patients to a group of 50 centinewton, 100 centinewton, 150, and 200 centinewton. Then after 1 month, we measure the amount of the tooth movement. At the same time, we measure the CGF, or crevicular gingival fluid, the amount of the activation of the biological proteins in that, and we compare them with together. Of course, to make sure this study is meaningful, we need to decrease the amount of the other variabilities. For example, the magnitude of the malocclusion should be the same. We decided to study class 2 D1 cases that require canine retraction, extraction of the first premolar. Biomechanic should be completely the same. We decided to compare bodily movement with each other. Remember, if we are using bodily movement against a control tipping or root movement, you are going to have a different response. And also we need to make sure that the patients are not taking any medications, following oral hygiene properly. In another word, any factors that affect the rate of tooth movement should be eliminated as much as possible. What we're going to find after 28 days? We notice if we're comparing the CGF protein concentration, the activity of the biological markers such as cytokines increase in response to increase to the magnitude of the force in the adult until certain level. After that level, the magnitude of the activity of this protein does not increase significantly. That tells us that there is a saturation point in human. This level for our study was about 100 centinewton, and we did not see the difference between 100, 150, and 200. How about the adolescents? Interesting enough, in the adolescents, we also see the saturation point, but in a higher level. In another word, there was a difference between 100 and 150 centinewton, but there was not a difference between 150 and 200. So this part of the study proved that there is a saturation point in human, and the saturation point between two groups of people can be different. Some group can have higher saturation point in comparison with the others. How about the osteoclast markers? Why that's important? The rate of tooth movement depends amount of magnitude of the bone resorption. Bone resorption depends on the osteoclast or bone resorbing cells. And let's see if we indirectly, through the markers of osteoclast, we can study the activity of these cells. Similar to the cytokines, we notice that the markers of osteoclast increase as we are increasing the magnitude of the force, and the saturation point of the osteoclast markers was higher in adolescents and less in adult. However, both reached to the saturation point after center level, and increasing the force was not able to increase these important biomarkers. How about the tooth movement? When we're looking at the trend of the tooth movement, the rate of the tooth movement increased with the magnitude of the force both in adolescents and in the adults. And adolescents shows higher magnitude of the tooth movement in response to higher magnitude of the force. However, this difference was not significant. That can be explained because we're having a 28 days study. During 28 days study, difference in tooth movement is not that much that can differentiate between the groups. This is not similar to the other markers such as cytokines and osteoclast markers that are more sensitive to our detection of the differences. You may ask why we did not increase the time to 56 days so we can study the tooth movement. As you are increasing the duration of your study, you will notice other variables started to creep in and they started to change the data. Based on this study, we can redefine our optimal force. That has been a concept that has been on discussion for a long time. Do we have an optimal force for all humans? No, we don't. The optimal force for each individuals can be different. We can define the optimal force, a force that reaches close to the saturation point, but we are not increasing it because saturation point is real, and increasing force does not necessarily increase the rate of tooth movement, just increase the side effect. I hope you enjoyed this session of CTOR channel. If you have not subscribed to our channel, please go ahead and subscribe, and please don't forget to press the like button. Thank you so much.